Hello everyone, my name is Prajakta Dhage and I am from Department of Zoology, KTHM College, Nashik. Today I will be taking a lecture for MSc Part 2. The subject is soft skill and the chapter is Unit 1. Topic is microscopy and this is the third lecture of the series. So today I will be talking about the phase contrast microscope. Now what is this? This is nothing but a special adaptation of light microscope which helps to obtain clear picture of living or unstained cells. Okay. Now, what is the specialization? The specialization is that it has adapters fitted in the light microscope. It was first discovered by Fritz Zernike from Netherlands. He was awarded Nobel Prize for Physics in 1953. Now let's see what is the principle of phase contrast microscope. So it basically converts the invisible small phase changes caused by the cell components which differ in their densities into the visible intensity changes. So it is going to convert the invisible small phase changes into the visible intensity changes. Okay. The phase changes are caused by the biological material through which the light passes. If the material is absorbent, it causes the waves to undergo change in amplitude and this is noticed by our eyes. The different hues of grays are noticed by our eyes due to the difference in the amplitude of the light rays. Now when the light passes through a living cell, they undergo invisible phase changes due to different refractive indices and also the thickness of the cell organelle. Now what is refractive indices or refractive index? It is defined as the ratio of the velocity of light in vacuum to its velocity in transmitted medium. Okay. Since the cell organelles are of different refractive indices, the light rays emerging would show variable phase changes. Okay. From this we can say that the behavior of light rays can be inferred as light ray undergoes a phase change depending on the refractive index of the transparent material or through the material which it passes. The phase change is directly proportional to the thickness of the material and light undergoes change in amplitude when it passes through an absorbent material. Thus, more the refractive index and thickness, more will be the change in the phase. Change in refractive index is also directly proportional to the thickness. Our eyes are able to notice the changes in amplitude which are perceived in the form of hues of grey. Okay. Living cells in general do not absorb light rays, but the cells and their components do show phase changes. The value of the phase change is 1/4 the wavelength of the light. This phase change is not noticeable to the human eye. The principle behind the phase contrast microscope is that it converts the unnoticeable phase changed into the noticeable phase change in the form of contrast and with the help of the two adapters. These adapters are called as annular diaphragm and the second one is annular phase plate. Okay. Now we will move on to the construction and working of the phase contrast microscope. Throughout the construction and working, you are supposed to pay attention to this particular diagram. Okay, then only you will understand how it works. So basically, phase contrast microscope is a light microscope with two specialized adapters. I have already discussed the annular diaphragm and the annular phase plate. Annular diaphragm is placed below the condenser. It consists of the circular disc and a circular annular groove. Through this, the light is allowed to pass. The light rays falls on the object through the annular groove. The image of this illuminated opening is formed at the back of the focal plane of the objective. Annular faceplate is placed at this particular plane. Okay? Annular faceplate is going to be of two types. One is negative faceplate, second one is positive faceplate. Negative faceplate has elevated thick circular area corresponding to the annular groove, and the positive one has thin circular groove corresponding to the annular groove. The thicker or thinner area in the annular faceplate is termed as conjugate area. The phase contrast is observed or obtained with the help of faceplate and the annular diaphragm by separating the direct rays from the diffracted rays. In a light microscope, condenser focuses light rays on the object. In absence of object, all the rays will be direct rays. Okay, there will not be any diffracted rays obtained. Okay, if an annular diaphragm is placed below the condenser, its image is produced at the rear focal plane of the object. If the eyepiece is removed, the image of the diaphragm is seen at the back of the objective. If an object is placed under the objective, some light rays will be diffracted by it. This diffracted rays will spread out and fill in the entire back lens of the objective and will overlap the central light rays. If the diaphragm is in the form of circular aperture, as we generally use in regular illumination, the degree of overlap becomes much more. Okay, and then it becomes impossible to separate two set of light rays. Okay, however, if the diaphragm has some special shape such as a slit or an annulus, the direct light will form as image of this annulus, but the diffracted light will be evenly spread over the whole back of the lens of the objective. Under these conditions, there will be some degree of overlap between the two set of rays, but this will be restricted to comparatively smaller area occupied by the image of the annulus. Okay, the phase contrast microscope also alters the phase and usually the amplitude of the wave in relation to other. This is achieved by inserting an annular phase plate at the rear focal of an annular groove. The working of phase contrast microscope requires no special skills or accessories. The image formed by the phase contrast microscope is due to the amplification of minor phase changes and their conversion into the changes of the amplitude. Okay, the light rays pass through the object, the undergo phase change and are different from the light rays which do not undergo change. After coming out of the object, the two set of rays pass through two different areas of the faceplate. The direct rays pass through either the elevated area or the groove area of the faceplate termed as annulus. The diffracted rays pass through the region other than the conjugate areas of the face. Now there is retardation of the light ray which passes through the biological material by 1/4 the wavelength of the light. This is a small retardation which cannot be detected by our eyes. The annular phase plate intensifies this retardation by additional 1/4 the wavelength. This intensification is either in the form of advancing or retarding the resultant rays. Okay, so this was about the working of the phase contrast microscope. Now we'll see the advantages of phase contrast microscope. So the main advantage is that it gives us the clear picture of the unstained or living cells. Okay, and without any prior treatment. This saves a lot of time, as very less or no preparation is required. It also prevents any kind of damage to the cells or any kind of alterations into the morphology of the cell. Okay, but although it has advantages, it also has some of the limitations. Now, the optical system of the phase contrast is such that the direct light rays falls on the annulus and the diffracted light rays on the entire phase plane. Thus, there is an incomplete separation of the two light rays, and this results in the formation of halo around the object. Another limitation is that the zone of action. Now, what is the zone of action? It refers to the heightening of the phase contrast effect near the edges and discontinuities. It progressively falls off towards the interior of the uniform object. Now, it is okay if you are restricted to the observation of cells, but if you want to do quantitative studies, this proves to be a bigger disadvantage. Okay. So, these were only the two disadvantages of phase contrast microscope. We will move on to the applications of phase contrast microscope. As I have already said it before also, phase contrast microscope is used to study living cells, and it is without any prior treatment or very lesser treatments. So, treatments like fixation is not there, staining is not used. Thus, we can study the cells in their normal conditions, and their normal structures in natural conditions can be studied. Some of the applications are that we can study the protozoans, we can study their different behaviors towards various physical and chemical treatments. Now, we can also study the behavior, mortality and other changes related to the living cells after the treatment with chemicals. We can study the cell cycle time, and we can determine the cell cycle time using the time-lapse cinematography. Then the processes such as phagocytosis, pinocytosis can be studied. Moreover, the permeability of the plasma membrane can also be observed by this particular phase contrast microscope. So, there are many of the applications to it, many advantages of it over the light microscope. Now, this was all from my side about phase contrast microscope, and thank you.